You know, deep down, every single living cell has one fundamental mission: to grow, get ready, and then divide. This process, it's literally the engine of all life. It's responsible for everything, from healing a scraped knee to the growth of an entire person. So let's dive right into this cellular mission and see exactly how life makes more of itself.
Okay, think of this image right here as our mission map. We're going to follow a single cell on its incredible journey, from the moment it's born, through this long, intense prep phase, all the way to that dramatic finale when it splits in two. This isn't just some boring diagram. It's the absolute blueprint for life at the cellular level. So before any cell can even think about dividing, it has to prepare. And I'm not talking about a quick last-minute check. No, this is a super extensive and critical period called interphase. It's where the cell meticulously gets all its gear ready for the big mission ahead. I like to think of interphase as the cell's ultimate training montage. First up, in the G1 phase, the cell bulks up, right? It grows in size and gets its metabolism fired up. Then comes the really crucial part, the S phase, that stands for synthesis. Here, the cell carefully copies its entire DNA blueprint. And finally, in the G2 phase, it does one last round of growth and a final systems check before the main event. And get this, it's kind of staggering. A cell spends over 95% of its entire life just in this preparation phase. The actual division part, that's just a tiny, tiny fraction of its existence. It's a pretty powerful reminder of how critical it is to get every single detail perfect before splitting. But, not every cell is destined to just keep dividing forever. Some of them, like certain heart cells or neurons, they finish their growth and then they just exit the cycle. They enter this non-dividing state called G0. You can think of it as a kind of cellular retirement. They're still active, doing their job, but they've hung up their division boots for good. All right, on to mission number 1, mitosis. The goal here is simple, but it's also incredibly profound. Create 2 daughter cells that are genetically identical, perfect clones of the parent. This is the process your body uses for growth and to repair tissues, like when you get a cut and it heals over. So, the action kicks off with prophase. I want you to imagine all that duplicated DNA. It's like long, stringy, messy spaghetti. And suddenly, it starts packing itself into these dense, tightly coiled structures we call chromosomes. This organization is absolutely essential for a clean separation. And at the same time, the cell starts building the machinery, the mitotic spindle, that's going to pull these chromosomes apart. Next up is metaphase. And this is just a moment of perfect, beautiful alignment. Every single chromosome lines up right down the middle, the equator of the cell, on what's called the metaphase plate. It's a supercritical checkpoint. Everything has to be in exactly the right position before the great separation can begin. Anaphase is the climax. It's fast, it's dramatic. In a sudden, decisive move, those spindle fibers contract and just rip the identical copies of each chromosome, the sister chromatids, apart. They get pulled swiftly to opposite ends of the cell. This is the shortest phase, but it's where the cell guarantees that each new daughter cell will get a complete set of genetic instructions. And the whole thing wraps up with telophase. A brand new nuclear envelope forms around each of the complete sets of chromosomes. Then, with one final pinch right down the middle, a process we call cytokinesis, the cell itself splits in 2. And the result? 2 brand new, perfectly identical daughter cells. Mission accomplished. But see, making perfect clones isn't always the goal. For sexual reproduction, life needs a completely different strategy. And that brings us to mission number 2, meiosis. This is a special ops kind of mission, designed not to create clones, but to produce genetically unique reproductive cells, like sperm and eggs. So let's just break down the key differences here. Mitosis, that's for your regular body cells. It's one round of division, and it makes 2 identical cells, each with a full set of chromosomes. We call that diploid. Meiosis, on the other hand, is only for making reproductive cells. It takes 2 full rounds of division, and it produces 4 unique cells, each with only half the number of chromosomes. We call that haploid. So how in the world does meiosis create all this variety? Well, it has a secret weapon. It's a process called crossing over. Early on in meiosis, paired chromosomes, one from mom, one from dad, they get real cozy and literally swap pieces of genetic material with each other. This is what shuffles the genetic deck, making sure that each of the 4 final cells is a totally unique combination of the parent's genes. The first big division, meiosis 1, is really unique.
This is where those paired chromosomes swap their genetic material and then separate from each other. This single step is what creates all that genetic variation, and critically, it's what cuts the chromosome number in half. The cell divides, and you end up with two cells, each carrying a unique halved set of genetic info.
Now, the second division, meiosis II, it actually looks a lot more like mitosis. In both of those cells that we just made, the sister chromatids are now pulled apart, and this gives us our final product, 4 genetically distinct daughter cells, each one ready for its potential role in creating a whole new organism. Okay, so we've looked at 2 very different cellular missions, with 2 very different outcomes. Let's do a little debrief and just summarize why each one is so fundamental to life as we know it.
So let's just boil it all down. Mitosis is all about consistency. It's about creating identical cells for growth and repair, making sure your genetic code stays stable. Meiosis is all about variety. It's about shuffling genes to create unique cells for sexual reproduction, providing the raw material for evolution. One ensures stability, the other drives change, and both are absolutely essential. You know, the cell cycle is a dance of incredible precision, with all these checkpoints and safeguards at every single step. But it isn't foolproof. And this raises a final, really critical question. What happens when this carefully choreographed process goes wrong? Because when a cell loses control of this cycle, the consequences can be devastating, leading to some of the most challenging diseases we face.